Welcome! Today we will explain to you the functioning and different types of agonists and antagonists. As you already know, our body contains many proteins that all perform different functions, enabling our bodily processes. These usually function by responding to the binding of other substances, called ligands, to their target sites. The activity of proteins is found to be tightly regulated, and this regulation is made, in part, via the binding of agonists and antagonists to specific proteins. Imagine a bouncer standing at the door of your favorite nightclub. This bouncer could be very lenient and let any person into the club. In this example, our bouncer is somewhat of an agonist. He functions to open the door to the club and allow people inside. Alternatively, a very strict bouncer could keep the door closed, leaving people on the street. We would call this bouncer an antagonist. This analogy is similar to what happens in a cell. Rather than a door, cells are lined with proteins. When the receptors of these proteins are unoccupied, the activity of the protein is low. This protein may, for instance, be an enzyme catalyzing at a very low base rate, or a channel protein, which remains closed when unbound. The binding of an agonist, the lenient bouncer from the example, however allows for signaling molecules to move into the cell or for upregulation of the enzyme's activity. The binding of an antagonist, the strict bouncer, keeps the door closed, the enzyme largely inactive. Without an agonist, the antagonist often has no visible effect, as the door, protein, was already in its inactive closed state. Still, the binding of an antagonist would then prevent the binding of an agonist until it leaves the binding site on the protein, functionally inhibiting agonists from activating the protein. Both agonists and antagonists are naturally found in our bodies, but still they may also be produced and administered as drugs in order to stimulate or inhibit certain processes. Some regulators are unique and are called agonist-antagonists. These can act both as agonist and antagonist depending on the conditions and environments in which they reside, for example in which tissue in the body. We have previously considered antagonists which bind to the same target site as the agonist, therefore blocking the target site competitively. Alternatively, non-competitive antagonists can prevent their target's functioning by binding at a different distant site, a so-called allosteric site, where it affects the target protein functioning either by physically blocking it or inducing physiological changes in the protein to prevent it from carrying out its function. To return to our example, a strict bouncer could limit the number of people coming into the club, not by directly being at the door, but rather by standing down the street and scaring them away. Up until now, we have presented antagonists as relatively permanent inhibitions of a protein, and for some this is actually the case. Insurmountable antagonists cannot be coerced into leaving their target's binding site and will remain bound for prolonged periods of time. Irreversible antagonists, for instance, may essentially be bound to their target forever, whereas other insurmountable antagonists leave the binding site eventually. On the other hand, even when bound to the protein, surmountable antagonists may be unbound when a large enough concentration of agonists is available. To come back to the example, if there are enough lenient bouncers to cover a shift, the strict bouncer would maybe also like to take the night off. If we go back to the agonists, different types of these may be identified. First of all, the so-called full agonists. The binding of these agonists elicits a maximum response, or 100% of its target protein function. The door in this case is completely opened. Different agonists can have different potencies, which can be due to the dose or concentration of the agonist or drug. Often, low receptor occupancy is already enough for a maximal response. For example, although only 10% of the receptors are bound, a complete response may still be produced. Of note is that the unbound receptors are not spare or inactive. These can still be bound by other ligands, like antagonists or partial agonists. This allows for lowering of the potency of the full agonist by binding of these ligands. A higher concentration of agonists is then required to again reach this maximal activity. As expected, since there are full agonists, we also have partial agonists. These molecules bind to and activate receptors to a submaximal response, even if all receptors are occupied. This means that partial agonists have a lower intrinsic efficacy than full agonists. Looking at our door analogy, partial agonists are only able to partially open the door. Partial agonists can also counteract the effect of full agonists, since they can compete for the same binding sites. Suppose a receptor is bound by a partial agonist. In this case, the receptor may not be activated or, to a lesser extent, reduced to full agonist potency. Next to full and partial agonists, there are also inverse agonists. To explain their function, it is essential to know that some receptors are constitutively active, so these receptors can signal in the absence of a ligand or agonist. This boils down to the fact that at a basal level, a significant number of receptors are active. In this case, the door will be always open. Once an inverse agonist binds to such an always active receptor, its activity decreases, the door is closing. As visible in these dose-response curves, inverse agonists have a negative intrinsic efficacy. These molecules have the same binding site, active site, as an agonist, so their actions can be reversed by a molecule with a higher intrinsic activity, a competitive antagonist, for instance. These agonistic and antagonistic features are used in pharmacology to design drugs that can act on receptors to elicit or block responses. An example of these are the superagonists, drugs capable of producing a maximal response greater than any endogenous agonist for that target receptor can elicit. These may also have a lower EC50, which is the dose at which 50% of the ligand's maximum effect is achieved. Superagonists often show an intrinsic efficacy of more than 100%. The door is opened even further than it would be by binding with an endogenous full agonist. The second to last type of ligand to be discussed are the physiological agonists. If we were to look at an agonist that has an effect when it binds to its receptor, and then observe another agonist binding to a different receptor, yet resulting in the same effect, we would call this ligand a physiological agonist. In fact, these ligands would both be physiological agonists of each other. A physiological antagonist would stop the action of an agonist, similar to a competitive inhibitor, without binding to the receptor the agonist uses. Examples of physiological agonists are epinephrine and hepatocyte growth factor, which both induce platelet aggregation by binding to their own receptors. An example of physiological antagonists could be found in one of the many substances that have antihistaminergic effects without actually binding to the histamine receptor. You can look at it as if there were two different doors to the same club, which are both used by a different person. Finally, we will discuss a certain group of agonists that, after they bind to their receptor, can favor a certain type of reaction over other reactions. In other words, an agonist can selectively activate a subset of a receptor signaling cascade. This phenomenon we call biased agonism or functional selectivity. The opposite of biased agonism is called unbiased or balanced agonism. Here, the ligands activate downstream signaling more globally without selectivity for specific downstream signaling cascades. Back to biased ligands, these work via stabilizing or inducing conformational changes or by changing the phosphorylation profile of their target protein in order to promote interaction with specific transducers, leading to the selective activation of downstream pathways. Moreover, allosteric ligands can also have biased agonism. This can happen in two ways. They may either act as biased ligands themselves, or when an allosteric and orthosteric ligand are bound at the same time, the allosteric ligand may interact with the ligand at the orthosteric site and promote activation of a specific subset of downstream signaling proteins. To visualize this, imagine a bouncer, in this case the biased agonist, opens the door, leading to a certain reaction. A differently biased agonist, a different bouncer, then opens the door, leading to a different reaction. We hope this video has refreshed or advanced your understanding of the different types of agonists and antagonists and their interactions with receptors. If we did, please remember to like, comment and subscribe to stay up to date on more of our content, which we'll probably release never.